Good day everyone, today we'll be discussing biotechnology. So biotechnology is defined as the use of living organisms or byproducts in the development of goods or processes that are beneficial to humans. So began several thousand years ago when humans began to domesticate animals and plants for the production of food. So since then many species of microorganisms and others like animals and plants have become routinely utilized by people. So more recently, it has become associated with molecular genetics. So in 1970, molecular genetic tools have provided novel ways to make use of living organisms for products and services. So recombinant methods also enable introduction of genetic materials into animals and plants, resulting to genetically modified organisms. So in 1980s, court rulings made it possible to patent recombinant organisms such as transgenic animals and so use of microorganisms in biotechnology. So microorganisms are used to benefit humans in various ways. So production of medicines, we have antibiotics, synthesis of human insulin and recombinant Escherichia coli. Then we have food fermentation like cheese, yogurt, vinegar, wine and beer, biological control, control of plant diseases, insect pests, and weeds. Then bioremediation is the cleanup of environmental pollutants such as petroleum, hydrocarbons, and synthetic. So many important medicines are produced by recombinant microorganisms. So in 1970s, it became aware of the great potential recombinant DNA technology to produce therapeutic agents for treating certain human diseases. So in 1976, Robert Swanson and Herbert Boyer formed Genentech Incorporated, which aspires to engineer bacteria to synthesize useful products such as peptides and polypeptide hormones. So Genentech's first contract was with researchers Keiichi Itakura and Arthur Riggs, and they were able to engineer a bacterial strain that produces somatostatin, a human hormone that inhibits the secretion of other hormones such as growth hormone, insulin, and glucagon. So they chose somatostatin because they thought it would be technically less difficult to produce than other hormones. So since then, recombinant DNA technology has been used to develop bacterial strains that synthesize several other medical agents. So what are the examples of medical agents produced by recombinant organisms? Of insulin, it promotes to lower glucose in our body. So it's treatment for diabetes. We have a tissue plasminogen activator or the TPA. It dissolves blood clots and used as treatment for heart attacks and other vascular conditions. We also have superoxide dismutase, an antioxidant, factor 8, a blood clotting factor, a treatment for hemophilia A. Then renin inhibitor, it lowers blood pressure, treatment for hypertension. Erythropoietin stimulates production of red blood cells, treatment for. In 1982, the US Food and Drug Administration approved the sale of the first genetically engineered drug, the human insulin, and was produced by Genentech and marketed by Eli Lilly. So insulin was isolated from pancreases removed from cattle and pigs. But unfortunately, diabetic individuals became allergic to insulin and has to use expensive combinations of insulin from other animals and human cadavers. So people with diabetes can use genetically engineered human insulin to treat disease. So we also have bacterial species as biological control agents. So it refers to the living organisms or their products to alleviate plant diseases. So non-pathogenic microorganisms are used to compete effectively against pathogenic strains of nutrients for space. So microorganisms may produce a toxin that inhibits the pathogenic microorganisms or insects without harming the plant. So an example is the Agrobacterium radiobacter to prevent crown gall disease caused by Agrobacterium tumefaciens. So the disease got its name from the large swelling or the galls produced by the plants in response to bacteria. So we also have Bacillus thuringiensis. Bacillus thuringiensis, usually referred to as BT. So it produces toxins that are lethal to many caterpillars and beetles that feed on variety of food crops. So BT is generally harmless to plants and other animals and humans and usually does not harm beneficial insects that acts as pollinator. Bioremediation is the use of living organisms or their products to decrease the pollutants in the environment. So during bioremediation via microorganisms, enzymes produced by microorganisms modify a toxic pollutant by altering or transforming its structure, an event called biotransformation. In many cases, biotransformation results in biodegradation, in which the toxic pollutant is degraded, yielding less complex, non-toxic metabolites. Microorganisms have been used in the treatment and degradation of sewage. So bioremediation has expanded into the treatment of hazardous and refractory chemical waste. So in 1980, recombinant microorganisms is patentable as a manufacture or composition of matter. The first recombinant microorganism to be patented was the oil-eating bacterium that contained a laboratory constructed plasmids. So this strain can oxidize the hydrocarbons commonly found in the petroleum. Unfortunately, as it has not been a commercial success because it metabolizes only a limited number of toxic compounds. Next, we have genetically modified animals. So the production of transgenic animals is a relatively new area of biotechnology. So gene replacement, so the genome of the host cell carrying the same type of the gene and clone gene is swapped with normal chromosomal gene by homologous recombination, leading to replacement of the normal gene with clone gene. Gene knockout, the clone gene has been rendered inactive and replaces the normal gene. Gene addition, the clone gene is introduced into a cell and becomes integrated into the genome by non-homologous recombination. So example of gene addition, production of aquarium fish that glow or the Glofish. So Glofish is a brand of transgenic zebrafish. Ang scientific name is Danio rerio that glows with bright green, red, or yellow fluorescent. Shi-Wan Gon and colleagues started with a gene from the jellyfish that encodes green fluorescent protein and inserted it to the zebrafish genome. So by placing the gene next to a gene promoter that would turn the gene on in the presence of environmental toxins, their long-term goal was to develop a fish that could be used to detect water pollutions. So researchers subsequently collaborated with a company to market fish for aquariums. So that is an example of Glofish. So they also developed red fluorescent zebrafish and yellow fluorescent zebrafish. So in 2003, Glofish became the first genetically modified organisms to be sold as pet. Again, Glofish. So genetically modified animals, so transgenic livestock may produce human proteins such as hormones in their milk. So this is called molecular farming, which involves the production of medically important proteins in the mammary glands of the livestock. So reproductive cloning and stem cell. Reproductive cloning refers to the methods that produce two or more genetically identical individuals. So Ian Wilmot and colleagues announced that a sheep named Dolly has been cloned using genetic materials from somatic cells. So this is the first cloned animal, Dolly. So in 2002, the first pet was cloned and was named Carbon Copy, also called as copycat. So mammalian cloning may potentially have many practical applications. It would enable farmers to use the somatic cells from their best individuals to create genetically homogeneous herds, which could increase agricultural yield. However, the disadvantage, it could be that genetically homogeneous herds may be more susceptible to rare diseases. So we have stem cells. Stem cells have the ability to divide and differentiate. They can be totipotent. It can give rise to all cell types. Pluripotent can differentiate into almost every cell type of the body. Multipotent can differentiate into several types, but far fewer than embryonic stem cell. unipotent can differentiate only in a single cell type. So stem cells have the potential to treat a variety of diseases. So from an ethical perspective, the primary issue that raises debate is the source of the stem cell for research and potential treatments, because most are derived from human embryos that were produced from in vitro fertilization and were subsequently not used. So agricultural scientists can now complement traditional breeding strategies with modern molecular genetic approach. In mid-1990s, genetically modified crops become first commercialized, and since then, its use has progressively increased. So what is GMO? GMO are products of a specific type of plant breeding where precise changes are made to plants' DNA to give in characteristics that cannot be achieved through the traditional breeding method. So we have golden rice, a rice modified with the daffodil genes to have more beta-carotene, which the body converts to vitamin E. Flavor savoir, tomatoes modified by removal of genes responsible for the softening the fruit, meaning the tomatoes spoil more slowly. The BT corn, corn modified with a bacterial insecticide gene so that it produces insect toxins with its cells, protecting it from pest species. We also have Aqua Advantage salmon, salmon modified with growth hormone regulating genes in order to grow to market sizes in significantly less time. Also have glow-in-the-dark animals, animals modified with genes for fluorescent proteins or glow-in-the-dark. This novel feature serves no practical purpose. So a very successful of the use of transgenic plants has been involved in the introduction of the genes from Bacillus thuringiensis. A molecular biologist can use the bacterium Agrobacterium tumefaciens to produce transgenic. So introduction of transgenic plants into agriculture has been strongly opposed by some because one potential risk is that transgenes in commercial crops could endanger native species. Planting of transgenic crops could potentially lead to proliferation of resistant insects. Despite these and other concerns, many farmers are still embracing transgenic crops and their use continues to rise.